So hello and welcome back all dear students again to another video tutorial by Genome Biotech. In this video tutorial, we are going to talk about the first class of molecules, class of signaling molecules which are present in a eukaryotic cell. And those class of signaling molecules include the GPCR, G protein coupled receptors that you can find, the ion channel receptor and the enzyme linked receptor. Firstly, we will be talking about the GPCR, that is referred to as the G protein coupled receptors. What are these G protein coupled receptors? How these G protein coupled receptors are being produced? In what manner these G protein coupled receptors are going to be produced? So that we are going to look forward. So before moving forward, let us look at analyze the GPCR. Where are these GPCR present? What are these GPCR and how the GPCRs are being arranged in our cell? So looking at a animation about the GPCR molecule. Okay, so this would be visible on your screen. Now starting the animation, try to understand what the Cooper says about GPCR. So GPCR molecules. Let us start.
A chain of reactions occurs in a liver cell when the hormone epinephrine binds to an epinephrine receptor on the cell surface.
Sorry, we are going to talk about the secreting molecules. Firstly, the first lecture that I have already told you in my yesterday's class, that what are these signaling molecules, how
There
signaling
are three
molecules are
general
present.
categories of signaling by secreted molecules, each based on the distance the signal travels. In endocrine signaling, hormones are secreted by endocrine cells and carried through the circulation to act on target cells at distant body sites. Only target cells with specific receptors react to the hormone. In contrast to hormones, some signaling molecules act locally to affect the behavior of nearby cells. In paracrine signaling, a molecule secreted by one cell acts on neighboring target cells. Only target cells with specific receptors react to the signaling molecule.
So only the target cell are going to receive the signal, and then they are going to induce a secondary response in the target cell. For example, the prostaglandins secretions from the localized cells which had certain injury, and those cells binds to a neighboring target cells. Next point.
In autocrine signaling, cells respond to signaling molecules that they themselves produce. These cells have receptors that bind to the signaling molecule.
So this is a type of autocrine signaling, and autocrine signaling was taking place in the case of cancer cells. Now, looking at the next animation about the GPCR molecules, what are the GPCR molecules and how does GPCR molecules are going to act? So let us have a look at the GPCR.
When an external signal, such as a hormone,
Okay.
binds to a receptor protein on the plasma,
Now, this is GPCR. GPCR refers to a complex of several different types of protein, the most important of which is commonly referred to as the G protein. This is the most important protein that is present in the case of GPCR. So, any protein molecule that has a G protein is said to be a GPCR molecule. Okay. If you want to understand about the GPCR molecule, you must pay the attention here. Okay. Now the GPCR could be categorized into monomeric GPCR or trimeric GPCR. Okay, so I'm going to write about both of these monomeric and trimeric GPCR molecules here in front of you. Okay, so GPCRs could be classified into monomeric GPCR and trimeric GPCR. I'm using this particular space. Do not get confused with that. Okay, so we are talking about GPCR. GPCR. A GPCR refers to as G protein coupled receptors. So GPCR contains always contain a G protein. GPCR GPCR refers to G protein coupled receptors. G protein coupled receptors in which a G protein is bound to a particular receptor in which a G protein is activated activated upon perception of a signal. Upon perception of a signal. So whenever a signal is going to be received, a G protein is going to be activated. Such type of molecules are called as GPCR, G protein coupled receptors. Now these GPCR can be trimeric GPCR, or they can be monomeric GPCR. Monomeric GPCR. What do you mean by trimeric GPCR? Trimeric GPCR has three different subunits. It has three subunits. Which are the three different subunits which are present? One is called as the alpha subunit, the beta subunit, and the gamma subunit. As well as there is a serpentine receptor. You can count that 1, 2, 3, 4, 5, 6, and 1 would be at the back. So there is a serpentine receptor. Serpentine means that it is going to transfer to the plasma membrane 7 times. Okay. This is how it is going to transfer. 1, 2, 3, 4, 5, 6, 7. This is how serpentine receptor is present. This receptor has been shown here to you. Okay. 3 subunit. Extracellular serpentine receptor. Extracellular serpentine receptor. Alpha, beta, and gamma subunits. 3 subunits. Which are the 3 subunits? The 3 subunits which are present as alpha, beta, and then it is the gamma subunit that is present. Monomeric GPCR. Monomeric GPCRs are the one which binds to only a single GTPase switch protein. These will never have the 3 alpha, beta, and gamma. These will have a single subunit equivalent to alpha. And these includes which are the 3 different types? The 3 different types include the RAS, the RAF, Rho, RUB proteins, Rho protein. The RAS proteins are all the different types of the monomeric GTPase switch proteins which are present in the cell. So these are different types of GTPase switch protein. Whenever I talk about the GTPase switch protein, whenever I'm talking about these GPCR as GTPase switch protein, GTPase switch protein. So this GTPase switch protein means that the protein becomes active only in the presence of GTP. They have GDP bound, hence inactive. Whenever the extracellular signal is going to bind, then the signaling will cause the GDP to be released and the GTP to be bounded. Then it will become activated. Okay. You can take a snapshot and I'm going to move forward. Okay. So I'm going to delete all these things and is now going to move forward. So let us have a look at the animation that how the activation of the GPCR, this is a trimeric GPCR, takes place.
Plasma membrane, the receptor molecule undergoes a change that causes a G protein to bind to it inside the cell. The activated receptor stimulates the G protein to release its GDP and bind to GTP, guanosine triphosphate. Bound to GTP, the G protein becomes activated.
Now, As the GDP has been hydrolyzed and the GTP has been bounded, the G protein has now become activated. As the G protein has now become activated, it will move and will bind to a specific effector protein molecules. Its binding to the specific effector protein molecule will activate the effector proteins.
The two parts of the G protein move away from the receptor and from each other to interact with target proteins. A target protein or effector may be an ion channel that opens or closes in response to the G protein, or it may be an enzyme that the G protein activates or inactivates. In any case, the effector produces a result inside the cell without the external signal ever having crossed the plasma membrane. When the external signal leaves, the receptor loses its activity.
So see this. The effector here refers to a molecule. The most important type of receptor which is present is the adenylate cyclase receptor. Adenylate cyclase. And this adenylate cyclase is a intrinsic plasma membrane protein which remains embedded within the plasma membrane. It is the one which will be catalyzing the interconversion of ATP into cyclic AMP. So ATP can get transformed to form the cyclic AMP molecule. Once the cyclic AMP molecule has been produced, so cyclic AMP will be expressed in the cell in higher concentration and will stimulate downstream signaling. It will catalyze downstream signaling. Downstream signaling. Okay, this is how downstream signaling occurs. And the downstream signaling takes place and large number of signals are going to be produced. Moving forward.
The alpha subunit of the G protein has a narrow window of activity. It soon turns off on its own by hydrolyzing GTP and thereby returning to its inactive state.
This is very important. As the author, Dr. Cooper, is saying that the GTP is being hydrolyzed. So GTP has been hydrolyzed, and there is an intrinsic GTPase activity of the alpha subunit. The alpha subunit has its own activity in order to hydrolyze the GTP to form GDP molecule. The GDP, once hydrolyzed, is going to move back and then it is going to prepare for the next signaling molecule to arrive.
The alpha subunit reassociates with the beta gamma complex, and the effector molecule turns off as well.
Okay. This is how signaling actually takes place in the case of GPCR molecules. This is the manner in which the signaling occurs in the case of GPCR molecules. Okay. Now, after the signaling of the secreted molecule signal transduction, the signal amplification takes place. Signal amplification refers to a cascade of series of reactions which are responsible for the amplification of specific signals to take place in the cell. Looking at that particular part, how the signal amplification takes place, how the G protein and cyclic AMP signaling occurs in the cell. So that we are going to look forward.
A chain of reactions occurs in a liver cell when the hormone epinephrine binds to an epinephrine receptor on the cell surface.
So this is equivalent to the section which is given in the book named Pathfinder, where you can find the different types of reactions that have been shown to you. Just moving to the book Pathfinder. So we are now going to talk about the first series of reaction in which the signaling molecule in the form of epinephrine is going to bind. So this is the epinephrine which is going to bind and is going to induce signaling via the alpha subunit binding with GTP and activating the other adenylate cyclase as well as the protein kinase, that is cyclic AMP dependent protein kinase, catalyzing the activation of the CRAB subunit. So the CRAB becomes activated and ultimately stimulation of gene transcription is going to take place. This is how stimulation of gene transcription is going to take place. Now, how it occurs, that we have to see.
The signal is amplified so that a single epinephrine molecule can trigger the release of many thousands of glucose molecules into the bloodstream. Each molecule of epinephrine activates a single receptor. However, each receptor may activate up to 100 G proteins, and each of these stimulates the enzymatic activity of one adenylyl cyclase enzyme.
Okay, see. So there is an activated receptor can stimulate hundreds of different types of GPCR proteins, and each GPCR protein has an ability to activate the adenylate cyclase enzyme. So this is the adenylate cyclase enzyme that will get activated. Once the adenylate cyclase enzyme will get activated, this activated adenylate cyclase will produce the next signaling molecule. How?
The next step in the cascade is amplified, considering that a single activated adenylyl cyclase may convert many molecules of ATP into cyclic AMP.
Okay, this is the
Multiple cyclic AMP
getting
molecules
converted
bind to the
cyclic AMP molecules. Okay, so the ATP is going to bind to cyclic AMP, converted to cyclic AMP molecule on perception of the signal via the receptor molecule and activation of the adenylate cyclase molecule. Once the adenylate cyclase has been activated, then the ATP will get converted into cyclic AMP molecule. Just now, the cyclic AMP is going to be transforming the other cellular target. So, looking at it,
regulatory subunits of the enzyme protein kinase A, resulting in the release and activation of its catalytic subunits.
okay,
Signal amplification.
this is how when the cyclic AMP is going to bind, the cyclic AMP binding will formation of the cyclic AMP will take place, and the cyclic AMP will bind to a specific proteins, and those proteins to which it is going to bind, those proteins are going to be referred to as the protein kinases. Once the binding to specific protein kinases takes place, the protein kinases will become activated. As the protein kinases becomes activated, then the next process of the protein kinase signaling is going to take place. How? Let us have a look at it.
Continues as each catalytic subunit of protein kinase A phosphorylates and thereby activates many molecules of phosphorylase kinase. Each of these enzymes, in turn, phosphorylate many molecules of the enzyme glycogen phosphorylase.
Now, protein kinase A is going to phosphorylate another targeted molecule, and that is referred to as the glycogen phosphorylase. Okay. Then the glycogen phosphorylase has become activated. Once the glycogen phosphorylase has become activated, then this is going to induce the activation of another molecule in the key process of signal transduction. How? Let us have a view.
Each glycogen phosphorylase may then trim off many molecules of glucose 1 phosphate from glycogen, resulting in the release of many thousands of glucose molecules into the bloodstream.
Okay, then it is going to induce the formation of the glucose molecule in the case of bloodstream. Just you can have a view or have a click about this particular diagram. Okay, you can just click on this particular diagram. The same is given in the book Cooper. We are also going to study this particular diagram, and its analysis is going to be done. Next, now what we are going to study is about this particular signaling molecule that is referred to as the G protein and the cyclic AMP molecules. Now, how the G proteins and the cyclic AMP molecules is going to take place, and in what manner the G proteins and cyclic AMP molecules are going to be targeted to the next step of the biochemical pathway that we are going to look forward, okay, through the book that is referred to as Cooper. Now, as I have already told you, the G proteins can be of two different types. One is referred to as the monomeric G protein, another one is referred to as the trimeric G protein. When you look at the structure of a G protein, you will find that these G protein molecules will have an N terminal molecule and a C terminal molecule present at one end and at the other end. The N terminal molecule contain the carbohydrate residues which are present. And these carbohydrate residues which are present over the N terminal molecule makes the receptor highly sensitive to a particular target molecule so that it can only bind to a particular target, particular signaling molecule. Then the signal is going to be transduced through the C subunit towards the target cell. See, if you look at the cases, The G protein or G protein coupled receptors. Whenever you talk about the G protein or the G protein coupled receptors, these are the largest family of cell surface receptor that transmit signal to the intracellular targets via the intermediate action of guanine nucleotide binding proteins or the G proteins. Today, more than 1,000 such G protein receptors have been identified. More than 1,000 G protein receptors have been identified, including the receptor for many hormones, many neurotransmitters. In addition, the G protein coupled receptors are responsible for smell, sight, and taste. The G protein coupled receptors are structurally and functionally related proteins characterized by several alpha helical membrane spanning domains. So these receptors which are present, they have certain key important features. The first feature of these receptors is that they are serpentine in nature. Serpentine means that they have traversed the plasma membrane 7 number of times. So these are serpentine receptors. If I can just show it to you all, just a second. These receptors are serpentine receptors. Sorry. Serpentine receptors. That is, they have 7 membrane spanning domain. 7 membrane spanning domain. Spanning domain, which is present. Okay, so these are 7 membrane spanning domains which are present here, and that is why they are said to be the serpentine receptors that you can find 1, 2, 3, 4, 5, 6, 7. Okay, the presence of these carbohydrate makes the receptor highly specific only to bind to a particular signaling molecule. Makes the receptor highly specific. Makes the receptor highly specific. Highly specific in nature. So this would be making the receptor highly specific so that it can bind to a particular specific molecule only. Now, these GPCR molecules are the main backbone of our sensory system. These serve as the backbone of our sensory system. The backbone of our sensory system. What do you mean by the backbone of our sensory system? Because in the absence of GPCR, we won't be able to smell anything, we won't be able to taste anything, touch anything, or we won't be able to even feel anything, as well as smell anything. So all our 5 system of touch, taste, smell, or visualization are all based upon the GPCR molecules. In the case of dogs, you can find dogs have approximately 3 times more genes encoding function for odorant receptor than in the case of humans. So dogs have 3 times more genes that are present for the odorant receptors than are the genes which are present in the case of humans. So as they have 3 different genes, 3 different types of genes for the odorant receptors, so they are much more sensitive to any sort of stimulus that might be present or that might activate them up. Okay, so dogs have approximately 3 times more genes encoding functional odorant receptors than are present in the case of humans. So this is very important. That is why dog can sense specifically what are the receptors which are present and how does these receptors are going to bind to specific molecules. The binding of ligand to the extracellular domain of these receptors induces a conformational change that allows the cytosolic domain of the receptor to activate G protein. So there is a conformational change that is going to take place in the cytosolic domain of these receptors. This is the cytosolic domain of this receptor. These are the cytosolic domains which are present. Now the cytosolic domain will undergo conformational change. Undergoes conformational change. Undergoes conformational change. as these undergoes is conformational changes. So these will be activating the receptor, different subunit. And the alpha subunit once gets activated. For example, this is the alpha subunit. Once this particular alpha subunit has gotten activated, then this alpha subunit will bind to the receptor molecule, bind to the target molecules. Okay, this is the target enzymes. These are the target enzymes which this alpha subunit is going to bind and transduce the signal further downstream. These are target uh, enzymes. Okay, once it binds to the target enzyme, it will be converting the ATP molecule into cyclic AMP, and the cyclic AMP is then going to induce the signal transduction to take place. Signal transduction. Okay, signal is going to be transduced, and once the signal transduction is going to take place, it will transduce the signal to specific receptors. Okay, so there are specific receptors. There are specific receptors to which the signal will be transferred. Okay, how the signal will be transferred, that we are going to look forward. This is how the GPCR has been discovered. Okay, these things are never being asked, but this one was perhaps the most important discovery. Okay, genome sequences have shown that mice as well as humans and certain other species such as dog has over 1000 odorant receptor genes which are present. Now see this, the G protein, as you know, is a heterotrimeric G protein. What do you mean by heterotrimeric G protein? Heterotrimeric G protein means that it consists of three different subunits present over itself. These refers to as the receptor, the alpha subunit, the beta subunit, as well as the gamma subunits, which are going to be present here. Okay. Now, there are the heterotrimeric G proteins. These are also referred to as heterotrimeric G proteins. As I have already told you that these are heterotrimers. These are made up of three different subunits. That is the alpha subunit, the beta subunit, as well as the gamma subunit. And there is a serpentine receptor to which a signaling molecule is going to be bounded. Now, see this. Heterotrimeric G protein, they are frequently called heterotrimeric G protein to distinguish them from other guanine nucleotide binding proteins. Now, there are different proteins which are referred to as monomeric GTPase switch proteins, and those proteins are RAS related GTPase binding proteins, as well as RUB, RF, RAN proteins, etc., which are present, which are responsible for several different types of transport proteins. For example, nuclear transport is under the hold of RAN. The vesicular transport is under the hold of ARF and RUB. Okay. And then the transport is going to take place. You can find here that this is how the activation of the GTPase switch protein takes place, and in what manner the activation takes place, that we must understand. Previously, firstly, what happens is that the binding of an hormone or the binding of a signaling molecule, a hormone or a signaling molecule takes place. Once the signaling molecule is going to bound, signaling molecule, once the signaling molecule is going to bind to the serpentine GPCR receptor molecules, this signaling molecule, when bind to the serpentine GPCR molecule, will induce a conformational change within the GTPase switch proteins such as alpha, beta, or gamma through either alpha or beta or gamma conformational changes, and will cause the interconversion of GDP with GTP. Okay, hormone binding stimulate the release of GDP and its exchange for GTP. The first step. Once the hormone is being bounded, the GTP is going to be attached, and the GPCR are going to be moving from an inactive state into an active state. Once activated, they will be inducing a conformational changes and will be targeting other enzymes as well as the ion channels. They will be themselves targeting the other enzymes as well as ion channel. The activated GTPase bound alpha subunit beta gamma complexes then dissociate from the receptor, interacts with their target molecules. Then they are going to interact with the target molecule. Activity of the alpha subunit is terminated by hydrolysis of the bound GTP, which is stimulated by RGS proteins. RGS proteins, the guanine nucleotide exchange factor proteins, are the one that will be again inactivating or preparing the cell for another round of cell signaling. This is how the inactivation is going to take place. First step, second step, the activated GTP bound alpha subunit and beta gamma complex then dissociate and interact with their target molecules. Activity of the alpha subunit is terminated by hydrolysis of the bound GTP molecule. Once the GTP molecule has been hydrolyzed, which is stimulated by RGS protein, the inactive GDP bound alpha subunit then reassociate with the beta gamma subunit. Okay, and preparing it for another round of cell signaling pathway. This is how cell signaling is going to take place. In the fourth state, in the inactive form, the alpha subunit is bound to the GDP in a complex which is referred to as alpha beta gamma complex. So this is the complex of the G protein that has been produced. Any doubt, any problem if you have, you can ask me up. Now, this is how the GPCRs are going to be bounded. Now, the cyclic AMP pathway and how does the cyclic AMP pathway works? The cyclic AMP, as the name indicates, is the secondary messenger and protein phosphorylation takes place. First, how the cyclic AMP is going to be produced. As the term cyclic AMP means, this compound has an internal cyclic structure present within itself. This is the internal cyclic structure which is present in this compound. You can find there is an ATP molecule, adenosine triphosphate, which contains 3 different phosphate. The first phosphate is commonly referred to as the alpha phosphate. The second phosphate is commonly referred to as the beta phosphate. And the third phosphate is commonly referred to as the gamma phosphate. Okay. Now, in the reactions to take place, the hydroxy group, which is present at the 3' carbon atom, this hydroxy group is going to attack the innermost phosphate atom. How? This hydroxy group is going to attack the alpha phosphate atom. As it is going to attack the alpha phosphate atom, so it will get bonded with the alpha phosphate molecule. Once it gets bonded with the alpha phosphate molecule, then it will lead to cyclization of this particular structure, leading to the formation of cyclic AMP. Once the cyclic AMP is being produced, the cyclic AMP can be neutralized by another protein called a cyclic AMP phosphodiesterase. This protein can influence the inactivation of the cyclic AMP and could lead to the formation of the AMP molecule. This is how this process occurs. That means when the signal is present, the ATP, that is activation of this adenylate cyclase, takes place through GPCR activation. Once GPCR activation occurs, then adenylate cyclase gets activated. And once the adenylate cyclase has gotten activated, then there is cyclic AMP phosphodiesterase molecule. And this cyclic AMP phosphodiesterase molecule is responsible for inactivation of the GPCR. It is the one which is becoming activated to cause GPCR inactivation. This is how GPCR inactivation takes place and will lead to the production of the molecule of AMP, that is referred to as adenosine monophosphate. So you can find here, this is the regulation of the glycogen metabolism by epinephrine. That how does the regulation of glycogen metabolism takes place by epinephrine. Okay, so you can look here in this part. I'm making this shorter so that only in a single diagram we can look here. Now, epinephrine is a hormone that binds to its own receptor molecule. Once it binds to the receptor molecule, it will activate the G protein molecules. Once the G protein has been activated, the G proteins will lead to the production of GTP molecules. Okay. The G proteins which has been activated, the GTP molecules are going to bind to are going to induce a signaling pathway to the alpha subunit. Once the alpha subunit has been activated, then the alpha subunit will bind to the adenylate cyclase, causing its activation. After the activation of adenylate cyclase has taken place, adenylate cyclase is going to transform the ATP molecule into cyclic AMP molecules. The transformation of the ATP into cyclic AMP will lead to the activation of the protein kinase A molecule. Once the protein kinase A has been activated, the regulatory subunit and the catalytic subunit of the protein kinase A are going to be separated because the cyclic AMP has the ability to bind to the regulatory subunit, causing the separation of the catalytic subunit to take place. Once the catalytic subunit has been activated, it is going to activate the phosphorylase kinase and is going to induce another pathway in which glycogen gets converted into glucose 1 phosphate and will lead to glycogen degradation. Glycogen degradation. Hence, release of glucose 1 phosphate into the bloodstream. Release of glucose 1 phosphate into bloodstream. Into bloodstream. This is how the release is going to take place. Okay. This is how cell signaling is going to take place. Now, similarly, you can find the same type of cell signaling being taking place here in the pathfinder molecule. Receptor binding causes the activation of adenylate cyclase enzyme. Adenylate cyclase will get activated. Once adenylate cyclase gets activated, adenylate cyclase will lead to the formation of cyclic AMP. Cyclic AMP will bind to the regulatory subunit. The catalytic subunits get separated. It will bind to specific molecules called as CRAB, which is referred to as cyclic AMP response element binding proteins. Once these proteins gets activated, it will stimulate the gene transcription to take place, and so the transcription of the genes are going to take place, and the genes are going to be transcribed, leading to the production of specific proteins. Hormone induced protein formation. Hormones induce protein biosynthesis. Induced protein biosynthesis. This is the protein biosynthesis that is going to take place. Okay. With these words, I'm going to make this class over. Okay. Just keep on revising the things. I'm going to upload your lectures and then we are going to meet tomorrow at the same time. Okay. Till then, goodbye and keep reading about GPCR as well as solve your MCQ questions. Good night. Bye bye.